Tu sors Julie. Tu sors Julie.
Yeah.
And you.
No, that's not that she's taking, and I cannot offer her anything but that. But yeah, like now I can see why I can go on extended delay. Yeah. Oh,
De là, je viens de mettre Reyes, Ordenas. She's from Russia. Now she's working on the structure of the team.
ah, yeah. So that was my project with Kathy, trying to verify the bloody protein. Yeah.
Ah, okay.
So yeah, no, so if she's working on that, that would be nice.
Okay.
Yeah.
I don't know how Greg communicates with the team.
No.
Trained for a long time.
Yeah, no. I tried that. I gave that. I had to take the cells across Montpellier, go to a plant by protein biochemistry institute at a friend of mine worked there, see the French place, get back to the bus and back to.
It's difficult.
Yeah, no, it's a pain. It's a joke thing.
Shall we go this week? I think we will start. So thank you everybody for coming. So today I'm very pleased to welcome Manuel Gonzalez Guerrero. So Manuel did his PhD in Granada, in Spain, working on heavy metal tolerance in arbuscular mycorrhiza in Conchi Ascon Lab.
Yeah.
Conchi Ascon Lab. Then he did the first postdoc in Guerrero, Canada, working on the distribution of metal in mycorrhiza in Larry Peterson's lab. Then did a second postdoc in Rochester Polytechnic Institute in USA, working on the structure function of metal transporting ATPase with José Arguello.
Yeah.
Then moved back to Europe with a Marie Curie Fellowship, working in Cathy Curie's lab, right, on iron homeostasis in Arabidopsis. And this is where he met the first time plants, I think.
A full plant, yeah.
Never too late.
Yeah, well.
And then he obtained Ramon y Cajal Fellowship for tenure track in Spain at the Polytechnic University of Madrid in Spain, working in metal homeostasis in plant microbe interaction. And now he's full professor over there in Madrid. And he will talk about his recent work on transition metal trafficking in biological nitrogen fixation. He was also a recipient of quite important grant, ERC and Bill and Melinda Gates Foundation grants.
Subgrant.
Subgrant. So
no, the real deal was Luis.
Okay, thank you very much.
So can you hear me? Okay, I have the tendency of talking too fast. So if that happens, someone please yell and I will slow down, hopefully. Thanks, Sylvain, for the invitation and the introduction. And it's always nice when they invite you to give a seminar somewhere, anywhere. But Toulouse is a place that really, I was very happy to be invited. I never worked here, but this is probably the city with the most influence in my career. So Guillaume Becard developed monosemic cultures of mycorrhiza that I used to do my PhD. So I look at Guillaume's work for many years. Then the work I did in the US with copy TPases. Well, the first guy who identified bacterial copy TPases was Jacques Bacchus, who also were here. He retired. Then most of the medical work that I'm showing, they come from the first 100 seats that Benoit Lefebvre gave me when I was a Marie Curie here. And thank you, Benoit. And also, you know, I'm going to talk about nodulation a lot, but we are not a lab of nodulation. I came very late to plants. So we are a metal group that happens to be working with nodulation. So many of the works that you guys publish here is something that we read, we learn your methods, we try to make them as good as possible, but it's, you know, it's a pretty good place to be here. So again, thank you for the invitation. As I said, we are a metal group. We are interested in transition metal elements, mainly, hold on, do you hear? Okay, well, we do it that way. Mainly iron, copper, zinc and nickel, and molybdenum mostly. The reason for that is because around a third of the proteins of a typical cell are metalloproteins. So that gives you a versatility. I'm trying to find with the bloody, yeah. So they give you versatility. So you can study almost any physiological process from the point of view of metals. And when you are setting up a lab, well, that's something that's neat. The other interesting thing about metals is that they are essential at very low concentrations. At the slightly higher ones, as the ones you can find in due to pollution in the industry. But here, do we have another pointer, someone? Because no, it's not the battery. I think it's broke down on the trip here. Yeah, it's not a pointer. Well, you have this here, right? Pointerless. So okay. So bear with me. So due to pollution or to the geological history of the site. So this is the Rio Tinto River in southern Spain, where it has high levels of copper and zinc due to it goes through different mine tailings and it's heavily polluted. And that's a problem because transition elements can produce non-enzymatically free radicals in femton cell reactions, and those they will damage DNA, they will damage proteins, and they will damage lipids. And on top of that, transition metals can also displace other elements from the active site of the enzymes, poisoning them. Or, for instance, copper can inhibit iron sulfur synthesis in mitochondria and chloroplasts. So controlling metals is something that's important. And actually, you can say that life is in a precarious age between metal toxicity, where you have oxidative damage and enzyme poisoning, and metal deficiency, where you have reduced energy production, reduced fertility, reduced response to hormones. And the equilibrium is so precise that there's less than 1 atom per cell. So that 1 free atom per cell means every metals are always bound to something. They're never pre-hydrated in the cytosol. That's what we were explained when they told us about the calcium ATPases, proton ATPases, and all those ions. So that's different. Okay, so I had to set up a lab. Plants, it had to be for a number of reasons. And for plants, metals are a huge problem. If you look at the high level, the main agricultural areas of the world, they have low metal viability. This is the distribution of zinc deficiency in soil. And as you can see, the American Midwest, the Mediterranean, the river valleys of China and India, for instance, they have a prevalent low metal viability that affects plant growth, plant yields, nutritional value. The 2 of the 3 main causes of malnutrition in the world are iron and zinc deficiency in those crops. So it's important for those plants to get the nutrients that they need to function, and for us to have those nutrients in our diet. On the other hand, if you look at more discrete points, you will see that, again, due to the history of the site or human activity, there are hotspots of metal accumulation in some soils, lead or cadmium, for instance, that they cause poisoning and they are damaging to the plant. So many groups have studied over the years how plants accumulate metals, how metals are uptaken from soil. Greg Vert did a lot of work on this, how they are translocated to the shoot, how they are used in different physiological processes, how they are transferred to the seeds, and they participate in almost every physiological problem. Now, luckily for me, when I was starting the lab, not many people had paid attention to the fact that plants do not live isolated, that they live in interaction with many bacteria. Some of them they are beneficial and some of them they are prejudicial, and that transition metals plays a role on that. So as in my PhD, I focus on arbuscular mycorrhiza that on top of adding phosphate, and they also act as buffers for metal nutrition. So if there's little available metal, they will improve metal uptake. If there's a lot of it, they will buffer it. And then we had to move to legume root nodules. And we were very interested in them because only 5% of the biomass of the nodule contains up to a third of the total iron content of the plant. and also very high levels of copper, zinc, and molybdenum and other elements. So as most of you know, nodules appear as interaction with between the legume roots and bacteria known as rhizobia. As a result of the chemical exchange that you analyze over here, this new organ appeared that contains the nitrogen-fixing bacteria that provide nitrogen to the whole plant. And recently, these cascades have been very well characterized, and the neem protein has been unveiled as to be one of the key regulators of the whole process. If you look at the nodule cells, you have these pseudo-organelles. These are the symbiosomes. These are the bacteria differentiated into bacteroids surrounded by a plasma membrane derived the symbiosome membrane. And here you can produce nitrogenase, which is the enzyme that converts nitrogen into ammonia with a lot of energy required in the process. So it has been known for many, many years that iron is essential for this process. If you grow the plants under low iron conditions, the plants grow worse. The nodules are white, they're non-functional compared to the regular conditions, where you can see the pink functional nodules. And lately, it has been, evidence has emerged that neem is actually affected by iron status of the plant, and it also affects the iron homeostasis of the plant. So there's also a link between nodule development and iron nutrition that sort of makes sense, because iron and other elements are essential cofactors of many of the enzymes involved in symbiotic nitrogen fixation. So the red color, that's due to the hemoglobin. That's an iron protein. That's 20% of the total protein of the nodule. It has a heme group, and it controls oxygen diffusion so that you can have nitrogen fixation in an anaerobic metabolism at the same time. Cytochrome oxidases use copper and zinc, copper and iron, and they bacteroids have a specialized one to work under the microaerobic environments of the nodules, and nitrogenase itself has 38 iron atoms and 2 molybdenum atoms per functional enzyme, and I think that that's the enzyme with the highest iron content, at least the one I can know of. So, okay, metals are important for this. We wanted to study them. We use as a model Medicago truncatula, mainly because they have this indeterminate growth pattern. So they keep the meristem, that's a zone 1. The nodule keeps growing over time. Then you have the infection differentiation zone, where the rhizobia comes from the outside and they differentiate into bacteroids within the symbiosomes. Then oxygen levels drop. You have the fixation zone. This is where the actual nitrogenase is working. And then if the nodules are enough, you have the senescence zone, where the nutrients get recycled. So it was a good model to study, because in one nodule you can trace the different developmental processes. You can study the different ways that metal enrich the nodules, and also we got someone to give us some seeds. When we started with a simple question: how do metals reach the nodule? So do they behave as roots, directly at taking the elements from the soil? Do they behave as leaves, as another zinc organ, so they are delivered from the roots to the nodules? Or do they have some sort of intracellular iron stores that they can use as they develop? So to figure out that question, we use a method, synchrotron-based X-ray fluorescence. So for that, we use a synchrotron, which is this equipment over here. This is Argonne National Lab. So it's a technique. Essentially, what you need is very high energy X-rays and very sensitive systems. You can bombard your samples with X-rays. The atoms will reach an excitation state. When they decay, they will fluoresce, and the wavelengths of the different atoms are different, and you can identify and trace elemental distribution within nodules, in our case, within nodules. So that's what we did. Okay, so let me walk you through this. So this is early zone 2, in this region over here. This is the differentiation zone. This is the vascular bundle. And we use calcium to define cell walls in red. We use zinc mainly to define nuclei in blue. And then we study iron distribution in green. And here you have the overlay. And what we saw was a dotted distribution around the vasculature. Then, as the nodule got older, you can see that the iron is sandwiched between the calcium-defined cell walls that we interpreted as being accumulated in the apoplast. And then when you go to the fixation zone, you see that the iron distribution is different. Now they are not between the cells, they are within the cells in this ring-like distribution. Then when we look at a higher resolution using another technique, nanosims, we can see that the iron co-localized with the carbon with the symbiosomes in that compartment. So what is actually happening, and these experiments and a number of others, show that iron and likely the other elements are incorporated from soil, directed by the vasculature into the infection differentiation zone of the nodule. Then they are released in the apoplast, and then a number of transporters get them from the apoplast into nitrogen-fixing cells and into the symbiosomes to synthesize all these enzymes. So, okay, that's what we wanted to study then. Many groups have worked on how plants incorporate metals, so we are not going to work into that. Legumes are dicots, so they know that they have a reductive strategy of converting iron 3 into iron 2. Then IRT1 and other transporters introduce them, and then symplastically and apoplastically, they reach the vasculature. But what we did not know at the time was how do they reach the nodule and what happens there. Okay, so we did the usual. The usual is you get mutants, you do RNA-seq, you have a good candidates, and you start screening. And one of the mutants that we first started work with was NAS2. This is a nicotinamide synthase mutant that grew worse under a symbiotic condition. Nicotinamide synthases are an enzyme that convert S-adenosine methionine into nicotinamine, and nicotinamine is able to chelate metals very tightly. And these nicotinamine chelated metals are used for radial transport and also for long distance metal transport along the phloem. And then the plants have specific metal nicotinamine transporters of the YSL family. And okay, we have that, so it sort of makes sense. Where is this nicotinamine synthase localized? And I don't think you can see it there. It might be too bright, but we found the localization of the protein in red around the pericycle endodermal layer of the vasculature feeding the nodules. I don't know if you can see it there. Anyhow, so we got the mutant. We studied elemental distribution at the synchrotron. This is wild type in this column, the mutant in this one, calcium and iron distribution. And as you can see, the mutants have way less iron in the nitrogen fixation zone. And consequently, nitrogenase activity in the mutant plants is reduced. And you can recover all these phenotypes when you are reintroducing the mutated gene. Not only that, we also got 2 mutant alleles of a YSL transporter, YSL3. This transporter is expressed associated to the vasculature in the nodules. They are in nodules, the vessels are in the periphery of the organ. And when mutated, they fix less nitrogen. And moreover, when we did, this is tabletop X-ray fluorescence studies. So the pictures are not as good and the method is not as resolutive. But what we can see is that they have differential iron and zinc distribution compared to the wild type. So this data and other, what showed is that metals are likely transported by the phloem into the nodules in complex with nicotinamine. And then you have a YSL transporter, YSL3, that introduce them into the endodermis pericycle cells. And for that, you need a nicotinamine synthase that provides this chelate. Similar work was done to identify a molybdenum transporter, MOD12, molybdate transporter, that also expressed in the vasculature, both in roots and in nodules, that we think it's mediating iron uptake from the vasculature. Nitrogenase also need molybdenum. Then we wanted to do the next step. How do they get released from the apoplast into the, from the endodermis into the apoplast? And unfortunately, we do not have a good answer for that. So if anyone has an idea, that would be nice. We know, well, so we do not know any transporter that is able to efflux molybdenum. A former postdoc in the lab, Manuel is working on that, but so far, there's no clear molybdenum efflux transporter in eukaryotes. And for iron, copper, and zinc, we do not have a good candidate based on transcriptomic assays. So there's nothing there, but it could be an HMA or VTL or NTP family member, but we don't really know about that. We have one weird anecdotic, so it's not published, but we thought it might be cool. Island-1 actually sees this. So we found an iron-specific transporter for reporting 2 that is expressed in the symbiosomes, but also localizes in the ER of endodermal cells. And we did electron microscopy because we wanted to know where it was, and we found in some images associated to vesicles, and some of the vesicles were also fused to the membrane. So that was sort of an idea that perhaps it could be that iron is not necessarily released through the plasma membrane, but through some exophytic process. We don't have any clear evidence except of what I told you about, but we like the idea because the amount of membrane that you have here is limited. The amount of iron that you need to provide is a lot. So by using the the endosomal compartments, you have more membranes to accumulate more iron and perhaps release more, but we don't know much about it. What we do know is that iron at the pH of the apoplast is not soluble. Iron will precipitate. And to keep it soluble, you need to bind it to something, and that something is citrate. In a collaboration that we did with Michael Utbardi's group a number of years ago, Michael identified MATE67, which is a citrate efflux protein, that when you mutate it, the nodules don't grow as well. They're actually really sick. And when we did X-ray fluorescence studies, if I can move it over here, yeah. This is the wild type. It had the similar distribution as before, but now in the mutant, you can see these hot spots of iron in the apoplast that we interpret as iron being precipitated because there's no citrate to keep it soluble. Then another group from Japan, Takashima, I think, if I remember correctly, also identified in lotus another citrate efflux at the endodermal level. So you need different transporters to keep it to keep the iron soluble in this context. Then we move to the bacteroids, and we wanted to know how, the infected cells. We wanted to know how iron, molybdenum, copper, and other elements got inside those cells. And again, we got 2 mutants, NRAM1 and MOD13. NRAM1 transported iron mainly. I don't know what they have done. Okay. So shall I do with that? So
The problem with Mac or?
okay. Switch it on and off, normally works. Okay.
Nice.
Okay, so here we are. How do I move to here?
If you go.
Thanks. Sorry. Okay, so as I was saying, we had 2 mutants, NRAM1. This is an iron transporter. How do we be my guest? You touch. Sorry,
It's okay. It's okay. It's okay.
Okay. So are we sure? No.
Yes, you have the.
Oh, there. Okay. So, as I said, iron transporter, MOD13 was a nodule-specific molybdenum transporter. We showed with promoter gas fusions that they were expressed in the fixation zone of the nodules. This was a bit later on as well, with immunolocalization that you can see in red. The distribution is in the periphery of the cell for NRAM1 and also for MOD13. You can see it, hopefully, you can see it there. And so that indicated that there were plasma membrane transporters introducing iron and molybdenum into nitrogen fixing cells. Okay, cool. But we also found transporters for other elements. For instance, we found a nodule-specific COPA transporter, COP1. So we only detected the transcript in nodules. The mutant was severely affected in nitrogen fixation, no phenotype when the plant were growing with ammonium nitrate. We know, and I always said that they transport iron, they transport COPA in this case, because we do GIS complementation assays. So we have a whole battery of GIS metal transport mutants. In this particular case, the CTR1 COPA uptake mutant. This one, since there's no COPA getting into bacteria, they cannot do oxidative respiration. And since they cannot do oxidative respiration, when you grow them with ethanol and glycerol, that you can only get energy through fermentation, the yeast will not grow unless you add a ton of COPA to the medium. But then when you put the Medicago truncatula COP1 transporter, you can see that there's a complementation of the phenotype. The COP1 is expressed in this interzone between zone 2 and zone 3, where we know that most of these elements have to get into the cells. And doing confocal microscopy, you can see that the red signal corresponding to the transporter is also in the periphery of the cell. We got the mutant. The mutant fixes 50% less nitrogen than the wild type. But then we have the question of, okay, but nitrogenase has iron and molybdenum. What's the connection with COPA? And the connection with COPA was the high energy requirements of nitrogenase. So nitrogenase requires 16 ATP per nitrogen fix. And if you remember your biochemistry from back the day, so ATP is provided by the ATP synthase. The ATP synthase needs the proton motive force that is provided by the electron transport chain in the mitochondria or the plasma membrane in bacteria. And for that, you need a cytochrome oxidase. That's a complex 4. That, as I said before, it's a COPA iron enzyme. So no COPA gets into the infected cells, no COPA gets into the bacteroid, and therefore cytochrome oxidase should be lower. And if it's lower, less ATP, and that's why you see a reduction of nitrogenase activity. So to prove it, we purified bacteroids, and when we purified the bacteroids, we saw a 50% reduction of cytochrome oxidase activity that we partially complemented when we added 10 times more COPA to the nutrient solution. So this and other data, what allows us to say is that, okay, iron 2 is provided by NRAM1, MOD13 provides molybdenum, COP1 provides COPA, and then I haven't shown you, but we also did work for zinc, and there's also a zinc transporter in this membrane introducing zinc. Now, problems. We know from the group of Javier Abadia in Zaragoza that citrates bind preferentially iron 3, but we know that NRAM transports iron 2. So that means that, as it happens in the plasma, in the epidermis of Arabidopsis, for instance, there has to be a ferroreductase that converts the iron 3 into iron 2. We could never find a candidate. So if someone happens to have the right mutant and you have that, send me a line, because I'm actually very curious to see which one is this one. But we are stuck there. Then we move to the next membrane, the symbiosome membrane, and we identified a ferroportin transporter, this one here, nodule specific, that introduces iron into the symbiosome. The group of Janneke Balk at the Young Innes, they identified another iron transporter of another family, a VTL8, that does exactly the same thing. We talked with Janneke quite a lot about this. So both of them do transport iron. Both of them are expressed in the same cell layers apparently. And if you mutate either of them, the nodules are white, they're non-functional, and so both of them are essential. They don't have any sort of redundancy that we could identify. So the only way that we hand wave around it is that perhaps you need so much iron there that one transporter is not enough. But why it was not a gene duplication? Why? We don't know. So no idea there. For molybdate, we don't know any molybdenum efflux transporter. Some people have proposed SST1. This is a nodule-specific sulfate transporter. So sulfate and molybdate, they have a similar structure. And some people say that perhaps it transports that, but okay, so but that's the idea. And then for copper and zinc, we have not identified anything. We think that it could be some P-type ATPases of the HMA family, but we don't have a good candidate yet. The other interesting thing about metal transport into the symbiosomes is that there are two efflux transporters in the symbiosome. So this was work done by David Day in Australia, by Alain Poupeau here in Nice, I think. And so when they first published this work, they proposed that the transporters were introducing the elements into the symbiosome. But that was because at the time we didn't have enough biochemical data on this family of transporters. So now the consensus is that they are actually transporting zinc and iron out of the symbiosome. And this has not happened during senescence. So what we think that they might be acting is as a pressure valves. So there's the idea is that we are adding so much iron here, so much zinc here, that we know that the bacteroids activate the metal detoxification systems because there's some risk of poisoning. And then perhaps you can recover that metal excess to prevent toxicity. But this is, again, a speculation. We don't have basis for that. What we are now interested in is to see how metals go from the transporters in the plasma membrane to the symbiosome membrane. Why? So I said at the beginning of the presentation that metals are not free, they're not hydrated. They cannot diffuse. So this is not like calcium. They have to be bound to something and transported to the other one. They have to be shuttled. We think that nicotinamine and other metal chelators, small molecules, are not doing that function. Why? Because you have hundreds of metalloproteins needing metals and only one or two transporters in the plasma membrane. So you need some way of giving preference to that. And what we think that is doing that is a heterogeneous group of proteins known as metallochaperones. So, and that has been very well characterized for copper. So as I said at the beginning of the presentation, metals are toxic, copper the same. They can produce free radicals, they can inhibit the synthesis of iron sulfur clusters. And to prevent that, the cells have developed a shuttling service. So this is yeast. In yeast has the CTR transporter that I use for the yeast complementation assay, and 3 copper chaperones. COX-17 that picks copper from the transporter and hands it for the synthesis of cytochrome oxidases in mitochondria. CCS1 that picks the copper from the transporter and gives it for, it's a copper provided for cuprocin superoxide dismutases in the cytosol. And then you have ATX1 that picks the copper from the transporter and gives it to a plasma, a P-type ATPase in the secretory pathway. And I was very interested in this family, ATX1, that they have this structure because it's something I was working with during my postdoc in the US. So these ATP copper chaperones, they coordinate one copper here in this loop, coordinated by 2 cysteines separated by 2 amino acids. And okay, we wanted to know what they were doing and how many Medicago truncatula had and how many Arabidopsis had, because there were only 2-3 papers published at the time. So we look at proteins in Medicago and Arabidopsis that had this domain, that they had the cysteine XX cysteine domain, and we found around 34 in Medicago truncatula. Fortunately, only one of them was nodule specific. And that nodule specific one had an N-terminal domain with a classical copper chaperone domain, a C-terminal domain that was intrinsically disordered, had 8 aspartates one after the other, only present in legumes, that we spent a year and a half trying to purify, and we were never able to produce from inclusion bodies. It was a nightmare. We recovered from the inclusion bodies, we added some copper, we diluted, we removed the guanidinium out of the way, and then precipitated again. So after a year and a half, Christina sort of gave up. She told me, okay, we focus on the N-terminal domain. We purify the N-terminal domain. This is nice. It can bind 1 copper per molecule, and it binds it with an affinity of in the picomolar range, which is what you can expect from a copper chaperone. We did the yeast complementation assays, and we know that it is a copper chaperone in yeast as well. So we went to the plant, And we did the usual. We check localization. The gene is expressed in this infection differentiation zone, early fixation zone. You have some expression also, but lower in the fixation zone. The protein was localized to those compartments as well. We did electron microscopy, and the arrows point at the gold particles, and we could find it in the cytosol, in the plasma membrane, and in the symbiosome membrane. So that was sort of compatible with an idea of picking up the copper from the plasma membrane, crossing the cytosol, reaching the symbiosome, and transfer it for nitrogen fixation. Now, if that's the case, we should see a phenotype under nitrogen fixing conditions. And that's what we saw. So the mutant grows worse. Nitrogen fixation is almost completely destroyed. But cytochrome activity is only, cytochrome oxidase activity in the bacteroids is only partially affected. It's not fully affected. We looked at copper content in these plants, and we didn't see any change in copper content in any of the organs tested in wild type and in the mutant compared to the wild type. And this was the first mutant in metal homeostasis that we had that did not show any altered metal homeostasis, any altered metal accumulation. And that sort of freaked us out a little bit. So we contacted Hendrik Cooper at the Czech Academy of Sciences. So Hendrik has a nice setup. You can, he has different HPLC systems where you can separate the proteins. At the end of it, you can collect each fraction you can divide in two. One you can use for metal content with ICP-MS. The other one you can use to do proteomics. So that's what we did with him. We had 2 biological replicates of the wild type, 2 biological replicates of the mutant, and by biological replicates is a pool of plants grown like 2 months apart. So that we try to space them as much as we can. Here you have the elution times, and this is copper content. And as you can see, some of the peaks are pretty the same, but you have this subset of the copper proteome that has lower copper content in the mutants and in the wild type. So it's not, all of this might dilute the effect, but then when you see only a small subset of the copper proteome, you can identify copper proteins there, but changes in the metallation of copper there. Since we could do proteomics, okay, we did that because we wanted to know the final copper acceptors of this metallochaperone. The problem, 2,500 candidates. So yeah, unfeasible. We were not going to do that. Then but we are stubborn, so we wanted to do it in a different way. So the PhD who did this work, Cristina Navarro, she always makes me say that she had to collect 10,000 nodules by hand with the help of other people in the lab. That's what I had because we do not exploit too much people. She got a protein extract. She passed the protein extract through a strep column. We wanted to know which were the proteins that were retained because we wanted to take them out in later analysis, but we were cleaning them of proteins that were binding to the resin. Then the flow through, we passed through another strep column that we had preloaded with the N-terminal domain, the copper chaperone domain of the protein bound to copper because we wanted to know proteins that would be accepting copper from the copper chaperone. We got dilution, we did proteomics, and again, over 200 proteins. Still less, but still too many. We crossed the data with the metalloproteomics studies, and well, now it became manageable. We got like 50. Many of them involved in some metabolism, some with peroxidations, and some blood proteins that we analyzed by other groups to have a role in nitrogen fixation. So okay, we started working on them. So you always have many false positives here. So we did a split YFP assays. Well, if you have interaction, you're going to see a green color. This is negative control. In some cases, you see the interaction in the periphery of the cell. This is done in tobacco cells, in epidermal cells, so you have them in the periphery here. In some other cases, you have them in the nuclei. And then later we found out that this protein that interacted with actually had a nuclear localization sequence. So it's bringing it to the nucleus. But again, it was not the best possible way to test that. But okay, we will try another way. So we use computational modeling with AlphaFold. You can play, and when we play, you can see that the copper chaperone sort of positioned the copper in a putative copper binding site with one of these interactor proteins that we purified and that we measured its copper binding affinity. So it could bind 1 copper per monomer with an affinity also in the picomolar range, but 10 times higher than the copper chaperone. So theoretically, it could be possible to recover copper from it. But we wanted to test that. So we did co-purification. So now we have the two proteins, the donor and the acceptor, one with a strep tag, the other one with a his tag. We can put them together, separate them in a strep column. When we do that, you can see that if there's interaction, well, the flow through, you always have the his tag protein. But if you have interaction, you will see some coelution. If you don't have interaction, or the interaction is too large, we will not see it there. But we went a step further. We added copper to the copper chaperone so that if you can see some copper being transferred to the protein in the flow through, you know that there has been some copper transfer in the process. So that's what we did. And here you can see that the copper chaperone coelutes with one of these interacting proteins. We showed that the flow through of this copper interacting protein contained copper when we incubated with the copper-bound copper chaperone. And although it's not shown here, when we separate these two proteins with a dialysis membrane so that the copper can get through, but there's no interaction between the proteins, no copper was transferred. So for the copper to go from the copper chaperone to the acceptor protein, you need protein-protein interactions. So these, and we are working on these other candidates and trying to figure out what they do. So actually, I didn't say that this USPA is a universal stress protein A. What it does, we don't know, but it is an nodule-specific protein. So it binds copper, and so we are going to we have the mutants, and we are now trying to figure out what it does. So again, we go to the model, and the model becomes a bit more complicated. So now we have some nodule-specific copper chaperones that get the copper from the transporter and gives it to the efflux protein. We are also working on iron chaperones that collect the iron from NRAM1 and gives it to for protein 2 on VTL8. But the data is at the moment a bit too preliminary to show. And we are also moving into the bacteroid. So, but working with bacteroids, it's a pain. Okay, so at least too complicated to purify, too heterogeneous the population. So we went for something easier. So we went for free-living diazotropes. We went for Azotobacter vinelandii because Luis's Rubio lab is in front of us. We started collaboration in this GATES project, so why not? So Azotobacter has two ways of uptaking iron: THEO-AB transporters and then metal siderophore complexes that I'm not showing here. We know that iron can be used to synthesize the cofactors of nitrogenase, but we know also that the iron can be stored in storage proteins in bacterioferritin, and those intracellular reserves you can use. So iron can go from here or directly from the outside. We know that synthesizing these cofactors is a nightmare. You need a bunch of other proteins to provide the iron sulfur cluster, to provide copper, to provide the homocitrate, to mature all these cofactors. And we were not going to get into that. We were interested in two proteins. NIFQ, because it's where molybdenum enters in the system, and NIFU, because it's a scaffold protein, where from iron and sulfur, you can end up with the 4 iron 4 sulfur clusters that directly or through further steps and maturation ends up forming the 3 different iron sulfur clusters that nitrogenase has. So we use bacterioferritin, we use THEO-AB, we use NIFU, and we use a bunch of other proteins as baits for co-purification assays. And we identified a number of interacting proteins. For instance, we know that NIFU interacts with NIFase. That's been known for decades. NIFase is a cysteine desulfurase, the enzyme that provides sulfur for the iron sulfur cluster that happens on NIFU. We found some reciprocal interaction with bacterioferritin, with NIFU, with glutaredoxin. This interaction was also interesting to us because it connects iron maturation for nitrogenase, iron sulfur synthesis, with the molybdenum entry into NIFU, into nitrogenase. So this is coordinating iron and molybdenum homeostasis. So we want to verify that. So what we do is, again, co-purification assays. So we showed, for instance, that NIFQ interacts with NIFU. So the two proteins that we saw, the entry of molybdenum and the entry of iron, they are interacting. We know that that interaction results in iron transfer from NIFU to NIFQ, and it does not happen when you have a membrane dialysis in between. And we know that what transferred is a 4-iron 4-sulfur cluster synthesized on NIFU, because we did spectroscopic assays, EPR assays, with Alex Guo, and according to Alex Guo and Carnegie, all these curves means that a 4-iron 4-sulfur cluster from NIFU got transferred to NIFU, and I trust him. This is beyond my expertise. So what we were able to position, so NIFU has been known for a long time that it can transfer 4-iron 4-sulfur cluster to NIFH. This is one of the 3 structural proteins of nitrogenase, the one that provides electrons, and also one that's involved in Fimoco maturation. Also provides a 4-iron 4-sulfur cluster to NIFB, which is the one that does the precursor of Fimoco. And now we show that NIFQ, it's also receiving a 4-iron 4-sulfur cluster from NIFU that then uses to introduce molybdenum for Fimoco synthesis for nitrogenase. We know as well that NIFU and NIFS interact because that's how you get the sulfur there, but we did not know how iron got into the system. And we wanted to study that. So NIFU is a funny protein. So a regular iron sulfur cluster is assembled in our mitochondria or in most bacteria by 2 proteins. You do ISCU, that's a scaffold protein. The electrons are provided by a ferredoxin. Ferredoxin has a 2-iron 2-sulfur cluster. And then the iron sulfurs are prepared here. They are transferred to a number of carrier proteins, such as NFUA, that they give it to final acceptor proteins. And this is present in Azotobacter. But under nitrogen fixing condition, they have this extra iron sulfur scaffold protein, NIFU, that it's the fusion in 3 domains of these 3 proteins. So you have the scaffold protein, the electron donor, and the carrier. But that also means that for NIFU to mature, you need 2 iron donors. You need an iron donor for the 2-iron 2-sulfur cluster at the catalytic site. You need an iron donor for the synthesis of the new iron sulfur clusters. So we went to the interaction map that we had. We started studying them. We verified some of this interaction. For instance, bacterioferritin, only in its solo form, only when it's loaded with iron, can interact with NIFU. And glutaredoxin, that has a 2-iron 2-sulfur protein, is able to interact with NIFU. Then we purified 2 versions of NIFU, the wild type one and a mutant in the cysteines that coordinate the iron sulfur cluster that is assembled on NIFU, not the catalytic one, the one that you prepare from de novo. And we did metal transfer assays. So let me go through these. So, okay, NIFU, when we purify, it's what we say control here. NIFU comes out with between 1 and 2 iron per monomer. That's the catalytic side. When we incubate it with bacterioferritin and we add DTT so that the proteins are released, the copper is released from bacterioferritin, we can increase iron, sorry. It can increase iron content in NIFU. So we thought that, okay, that means that bacterioferritin can act as an iron donor for iron sulfur cluster synthesis, but we need to do a few controls. One control is that when we use a dialysis membrane in between, that's a what's control, there's no transfer of iron. So you need, again, to have protein-protein interactions. The other control that we use is we use this version of NIFU, the one that cannot assemble a de novo iron sulfur cluster. And when that happens, the iron does not significantly increase in that line. So that means that bacterioferritin will give iron to NIFU for iron sulfur cluster synthesis de novo. With glutaredoxin, we did something similar. So we regenerated the 2-iron 2-sulfur cluster on the protein. We use this mutant version of NIFU, and we incubated it with EDTA. So we tried to remove as much as possible from the iron sulfur cluster that we could. It's not perfect. We only got probably 50% of the total iron content, as it was there. But when we incubate it with these 2 proteins together, we separate them, we can see that the iron content in this mutant one increases. So we think that we can interpret this, and it's a bit preliminary yet, as a way of transferring iron to the catalytic site at the core domain of NIFU. But this, we have to work a little bit more on this. And why this interest? So this comes into context of this Bill and Melinda Gates project that there are different projects interested in getting nitrogen fixing crops. Luis Rubio's ones, the ones I participate in, his goal was to express all of these nitrogenous components in mitochondria and chloroplast so that the plants on their own can produce functional nitrogen. So that's a dream. So, and that means getting around 10 proteins working there, metalloproteins, and to synthesize all these metal cofactors. And they have been quite successful on getting them, producing them in plants, and being able to work and showing that those proteins can work in vitro when they are purified. The problem is that the yields are very low and the metal content is very low. So for some reason, when you are expressing those proteins in mitochondria or in chloroplast, we are not plugging them to the metal homeostasis networks in bacteria, in plants. And that's what we are doing now. And that's why we are interested in how Azotobacter provides iron and molybdenum for the synthesis of Emoco, because we have to transfer that as well. So that's what we have been doing. So this is bacterioferritin expressed in Nicotiana, Nicotiana tabacum. Here you can see it with GFP that they colocalize to chloroplast because we direct the proteins with signal peptides to this compartment. We verify them with electron microscopy using a twin strep tag protein, and although you might not see it here, well, you are not going to see it because I have a hard time seeing it. But there's gold particles associated to the chloroplast, so it's getting there. This has been done by Luis's group for many years. So they showed that NIFU, this scaffold protein, they can produce it in chloroplast to some degree of purity with yields around 50 milligrams per kilogram of leaves, but with very little iron, only 0.5 iron atoms per monomer. And the functional one should have at least 2. So this means that 25% of the protein is only working, all the rest is useless. So when we co-express with bacterioferritin, one of these interactors, we have beta purity, probably because we have increased the yield, so we have less proportion of contaminants. We double the amount of protein, but we do not have much success in getting more iron there. However, when we combine that with the glutaredoxin, the one that provides a 2-iron 2-sulfur cluster to the core domain, the yields do not improve significantly, but this is N1, so this is still preliminary, but highly encouraging. We can almost have 100% of the protein fully functional. So by combining the two things, we have gone from 25% of the protein active to almost 100%, and also doubling the amount of protein that the plant can synthesize. And we have seen something similar at the yield. We haven't done iron content yet, also with NIF-H and NIF-B. So in this model, again, we now have 2 other elements, glutaredoxin that provides a 2-iron 2-sulfur cluster to the core region. At least that's what we think now. And bacterioferritins that provide the iron that they are used to synthesize the 2-iron, the 4-iron 4-sulfur cluster synthesized on NIFU. And this was going to be the presentation, but I added one extra slide really fast on a side project of the lab. But it has nothing to do with nitrogen fixation, but it has to do with a zinc-mediated immunity. So we got some internal co-funding to do collaborations among the different labs in the institute, and a good friend of mine, she works in plant pathogen interaction. We had been time in Switzerland, so hey, let's get some Arabidopsis leaves. Let's put the pathogen she was working with. This is a Plectosphaerella cucumerina, a necrotrophic fungus. And let's see what happens with elemental dissolution. And what she saw, what we saw there, if I can put it here, is that, well, you can see the spot here, that's a mold. There's more calcium, that should be expected, but also more manganese and more zinc in the infection site. So, okay, that's neat. It could be intentional or something accidental. So we use a doubled mutant in Arabidopsis, the HMA2, HMA4 mutant. That's 2 zinc ATPases that Arabidopsis has that are involved in long distance translocation of zinc. And when you have that mutant, well, iron accumulation does not happen. And recently, in collaboration, like 3 weeks ago, in collaboration with Anthony van der Rent, we went to another synchrotron in Hamburg. And we wanted to know which were the molecular patterns eliciting this zinc accumulation. So this is MOG, and these are different microbial associated molecular patterns and damage associated molecular patterns. And as you can see here, there's accumulation. You don't need the whole fungus there, only simply some molecules to elicit that response. And actually, yeah, I think that technology is fantastic. So these took like 4 hours to make a leaf. Yeah, the synchrotron, this took like 20 minutes, like ridiculous. I think that amazing now. And we also look at the zinc distribution at higher resolution, 2 micron, and I don't know if you can see it with the beamer here, but zinc is in the periphery of the cells, it's shaping the epidermal cells. So we think that it's being translocated to the apoplasm or to the plasma or to the cell wall to combat the pathogen. So finally, so some messages. So symbiotic network fixation requires dedicated metal delivery systems. And that, if you are interested in metal homeostasis, makes nodules a really good model to study metal transport and metal nutrition. Because you lose most of the functional redundancy that you have in other plant organs. You can use it to identify new metalloproteins, and using metallochaperones as bait is a good way of finding out what are the downstream proteins. And then you need to understand, or we need to understand, cytosolic metal transfer in diisotrophic organisms if we want to have functional nitrogenase in plants. And this is the current iteration of the lab. This is the funding, and we like collaborating with people. It's part of the fun. You get to learn a lot of people. Actually, this is not an extended list, but I'd be happy to take any questions, and thank you for your time.